Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Maxime Dumont from PolyPlus and Emmanuel Camus from Paul Corporation.
Hello everyone, and thank you very much for joining us today for this webinar. The idea today will be to give you food for thought related to bioprocess economics for viral vector-based gene therapy. So we will begin with a short introduction about PolyPlus and PAL, and then continue related to the therapy affordability to give you an overview of the cost for an AAV-based gene therapy. And the most interesting part will be to discuss about the critical parameters to influence the cost per dose, and then we'll give you the key leverage to actually reduce the cost per dose. And we'll conclude on how PAL and PolyPlus can help in the bioprocess economics. So let's start it with PolyPlus. So we are a French biotech company located in Strasbourg near the German border. Our core business is around transfection. So we are providing transfection solutions for cell and gene therapy to produce viral vector AV or LV. We also provide alternative to viral vector use, so which is actually the in vivo transfection reagent that can be used directly into the patient to transfect both DNA or RNA therapeutics for cancer therapy or even vaccination. Transfection reagent can also be used for protein production and also life science. So we consider PolyPlus as a key actor of process economics because we are providing components of the upstream manufacturing. So basically a transfection reagent up to GMP pharma grade. And we recently acquired EasyVec, which is a company focused on the engineering plasmid. So we can provide now this kind of service at PolyPlus. So now Emmanuel Camus will give you an overview about PAL Corporation.
Hello, everyone. Very happy to be here. So PAL is a part of the Danaher Corporation since several years now. And here's an overview of the different segments within the Danaher Corporation. So we stand within the life science division, where some other sister companies you might be familiar with, such as Cytiva, Precision Nanosystems, and more recently Aldevron, along with Beckman Coulter and Leica. So we're really part of this multi-industry science and technology portfolio. Regarding PAL, so PAL provides integrated solutions for biotech applications. And our aim is to serve the complete process from the media preparation, buffer preparation with our single-use mixers and storage solutions and filters towards the connection, disconnection of single-use manifolds. And then going to the process with bioreactors for let's say bench scale for either adherent with our multiple bioreactor or suspension with our biaxial 25-liter bioreactor towards the cell culture suspension and adherent large-scale production, the cell harvest towards all the downstream with buffer management systems, filters, storage solution, purification solutions, TFF chromatography, and all the fill-in finish technologies.
So now let's discuss about the therapy affordability. So we all know that the cell and gene therapy has a bright future because we are touching DNA. It brings unlimited possibilities of treatment. So we took two examples here of AAV gene therapy. So Luxturna, in one hand, this is a treatment for ophthalmic disease, and Zolgensma, in the other hand, to treat the spinal muscular atrophy. So you can see that the cost per dose is pretty different from a drug to another. So Luxturna is around $850K for both eyes, whereas the Zolgensma is a 2.1 million single dose, and it represents actually the most expensive drug product of the world. So the reason behind, to my mind, is the lack of maturity of the bioprocess in a way where we need to produce more and we need to produce better quality of AAV. And for that, we need adapted technology that Polyplus and Paul can supply. So the main question remains, which is actually related to the therapy accessibility, because if we are developing treatment, this is actually for patients, and as the cost is really expensive, we need to have a healthcare program to access those drug therapy. So we can say that there is a part of the population that will not be that would not have access to those drugs. So the main question for us is actually how could we reduce the cost per dose? So the industry is evolving quickly by innovating the way we produce. All the suppliers are providing technology to improve productivity, but it cannot be a success without the help of the user of our product. So collaboration between producer and supplier is a key factor of success. So here I give you an overview about the cost of an AAV-based therapy. So you have in one hand the OPEX that regroup both consumable, reagent, medium, sales flask, but also salaries. So the OPEX represents 30 to 40% of the manufacturing cost. And in the other end, you have the CAPEX, which represents 60 to 70% of the manufacturing cost, and it includes equipment such as fixed bed bioreactor, so ICLs. You have also side tank bioreactor, mixer, incubator, and so on. And include also the building, which is actually the major part of the cost of related to the CAPEX. So on the right, we have a focus on the consumable part. You have the proportion of the cost of goods for a single 100-liter batches using PI Pro. And you can see in this graph that the plasmid, for example, represents 30 to 40% of the proportion of the direct cost of goods. We can also say that the transfection will affect the quantity of plasmid. So basically, we can say that optimizing transfection condition to find the best balance between consumable and productivity will affect the cost per dose. So what would be the impact of increasing the functional AAV productivity? So first of all, if you increase productivity, you increase the number of doses at the end. So you can treat more patients per year, but it will also affect the cost per dose, and we will see that in the next two slides. If we take the example of two different processes, one with PI Pro transfection reagent and the other one with Vector-V AAV transfection reagent. You have the whole batch, similar consumable, you just change the transfection reagent. So if we say that to reach a specific target in terms of quantity of our vector, with PI Pro, you need, for example, 6 batches to reach this target. With Vector-V AAV, as it's twice more productive than PI Pro, you need to use twice less batches to reach the same quantity of viral vector. So instead of having, for example, 6 batches with PI Pro, you need only 3 with Vector-V AAV. So it reduces not only the transfection reagent, but it will affect also all the consumable related to a batch. So medium, cells, but also filters, chromatography column, etc. If you see it differently, you can also decrease the bioreactor size. So instead of decreasing the number of batches to reach the same quantity of viral vector. So if, for example, with PI Pro, you have a 100-liter bioreactor, you will have the same quantity at some point with a process with Vector-V AAV using a 50-liter scale. So this means both situations, so reducing the batch or reducing the bioreactor size, will affect both OPEX and CAPEX, and it will touch also the manufacturing footprints, which represent the major cost of the manufacturing process. So for me, there is two critical parameters to influence the cost per dose. The first one is the scale. So you can see the correlation between the number of doses and the cost per dose. So if you increase the volume to produce your dose, of course, you will increase the number of doses. So the more you increase the scale, the more doses you will obtain at some point. And you will also reduce the cost of the dose. Why is that? Because for each specific batches, you have a fixed amount of parameters, so salaries and QCs, for example, you have the same expense dedicated to quality control if you use a 50-liter bag than if you use a 2,000-liter bag. You will have to perform the same quality control on the same number of doses, let's say. So you will dilute this fixed amount, for example, over the scale. So the second parameter is actually the productivity. So you have to optimize your upstream process to have the better productivity possible. So if you compare again Factovie AAV GMP and PI Pro, you can see that the more you produce, the more doses you will have at some point, and again it will affect the cost per dose. So let's illustrate this into a theoretical case study. So in which we compare three different processes: Factovie AAV, PI Pro, and another PI. If we take an average dose per patient that corresponds to a Zolgensma dose, so 8.8 10 to the 14 VG, so it's a total quantity of viral vector. The productivity comes from in-house experiments. So with Factovie AAV, we've got 4.8 10 to the 11 VG per ml. We've got twice less with PI Pro and about 6 less with another PI. And you can see that in the fourth line, this is the volume of culture needed per dose. So to achieve one dose, how many liters you will need to have with Factovie. So this is 6.1 liter. With PI Pro, well, it's around twice more and around 6 more time for the other PI. So you can see that, again, if you reduce the volume of the bioreactor, you reduce both OPEX and CAPEX. Then, if we have a situation in which we would like to produce in a 2,000-liter bioreactor, how many doses you will get? So with Factovie AAV, we have around 300 doses. With Factovie AAV, with PI Pro, it's around half less, so 160. And with the other PI, it's around 50. So if you want to reach a specific target in terms of number of doses, so here we have 100 doses produced in a 2,000-liter scale. So we need 4 batches with Factovie AAV, 6 batches with PI Pro, and around 20 for the other PI. So again, the performance of the transfection reagent will affect the number of batches, and the number of batches will have a drastic impact on the manufacturing cost and the cost per dose. So now Emmanuel will give you an example about the performance of our reagent into an iCELLis Nano.
So as Maxime was explaining, the differences in reagents and transfection reagents chosen can have an impact on cost, and because they have an impact on performance. So in order to illustrate this, we recently conducted a study together with a customer in Vitria. So this has been published at ISCT recently, where they performed an assessment of different transfection reagents in the iCELLis nanobioreactor. So HEC293T cells were transfected with a single GFP plasmid, and the output of this study was that all cultures transfected with the different transfection reagents had similar metabolic profiles and therefore similar cell growth. But when they, after transfection, they sampled some fixed bed strips from the different regions of the nanofixed bed in order to assess the transfection efficiency of the cells within the bed. The cells were lysed and the fluorescence was measured in order to assess the productivity and the transfection efficiency. And so you can see here on the graphs that are on the right of the screen. So the graph on the left compares normalized GFP for PI Pro supplemented with optiferin and a liposome reagent. And you can see that PA Pro in this process gives better results than the liposome reagent, whether it is in the top, in the middle, or in the bottom of the bioreactor. Whereas on the right, they have compared PI Pro with supplemented with optiferin again, with Vectovir AAV supplemented with optiferin as well. And here as well, we see that for top, middle, and bottom areas of the bioreactor, in this case, Vectovir AAV provides average of double productivity in GFP than PI Pro. So we can see significant differences in the productivities with different transfection reagents on the same exact process. And therefore, it is really recommended in order to reduce the cost of the dose and the cost of the overall process to screen reagents, and the transfection reagent is one of them, to find the best fit for your process. Some other information around this and related to what Maxime was explaining earlier, as cost modeling can be really a tool to evaluate the benefits of these different alternatives on the overall process cost and cost of those. So this is a study that has been presented in 2020 at ASGCT, looking at how cost modeling can help understand the major cost drivers and impact of technologies processing choices on AAV production. So it looks at comparing traditional multi-tray process with a suspension process and with an isellus adherent bioreactor process. So here you see the different assumptions that were made on the right, because every cost modeling needs to come with a certain set of assumptions that certainly define this. And of course, as soon as one assumption is not true, the output is not anymore valid, let's say. So this needs to be kept in mind when looking at the data. So the downstream process is common to all platforms, and you have, so here on the left, the different processes that were modeled in this study. So one of the outputs of this study is looking at the different cost split, let's say, of the different parts of reagents and manpower and consumables. And we can see that the GMP grade pDNA and the transfection reagents are a common cost driver in all processes. On the table on the bottom, we have the batch cost. So certainly when we work with an optimized suspension or an optimized isellus, so optimized here means an optimized pDNA ratio per million cells. We certainly get a reduced dose cost. The number of batches per year in the isellus are a bit lower here, because the bioreactor, the part of the model assumes that the bioreactor is used for a longer time because we seed lower than in suspension. So basically, we have a longer immobilization time, which reduces the number of batches that can be done per year at maximum capacity. But I mean, in globally, this data shows that really there's importance in optimizing the pDNA use, whatever the platform is chosen, and part of that is choosing the right transfection reagent. So looking at increasing productivity, production efficiency, we also need to think of what is their impact on the downstream cost. Because on the one side, when we think of increasing the productivity on the upstream, we will not increase only our product of interest. We will probably increase contaminants, whether we will increase also process and product-related impurities. And this needs to be balanced with the filter capacity, sizing, the chromatography support capacity and sizing as well, and the number of downstream steps that will be required to remove all these extra contaminants. Because of course, if you win certain things on the one side, that might be allowing you to decrease the cost, but on the other side, you need extra cost to eliminate all these extra contaminants, then the balance might not be exactly there. So it is really important to optimize the downstream alongside the upstream. So together, when changing things on the upstream, you immediately need to take a look at what is the impact on the downstream in order to optimally optimize the process and the overall cost of those. Because so basically the cost decrease of the productivity increase needs to be larger than the extra cost that you will have on the downstream.
So for me, there is 4 different leverages to reduce the cost. The first one is the choice of the manufacturing model. So is it internal or do you want to use a CDMO? It basically depends on how many products you have to manufacture, and it depends also on your own manufacturing capacity. If you would like to produce it by your own, you need to think about the building, because the building will build the process will build the product, and you can also reverse it. The product is the process, and the process is the building. The other leverage is actually the consumable, because the upstream will lead the productivity. So you have to carefully choose your consumable that works in synergy. So basically the cell, the transfection region, the plasmid, the medium, everything has to be optimized during the process development phase to optimize both productivity and scalability. Then you have the choice of the equipment. So this is based mainly on the scalability and the robustness of the process. So the objective of the upstream is really to optimize the productivity, but behind it, there is the downstream process. And the idea of this downstream process is to avoid losing too much functional titers. So as Emmanuel said, this is about balance the upstream productivity and the quality of the upstream productivity with the downstream process. It's also very important to really understand your process from beginning to the end. So meaning that the analytical part has to be carefully done, and the process has to be monitored and controlled. The last leverage for me is the choice of the supplier, because you are not only purchasing a product. So the product has certain quality for sure, but it's also about the supplier. The supplier can help you in designing your, let's say, optimizing your experiment. We have also a regulatory support that could help also to accelerate your time to market. And as you may know, time is money, so we really need to ensure that your drug approval is good. So how can Polyplus help reducing the cost? Basically, we are providing transfection reagents that have a key impact on the productivity and on the scalability. So both parameters are the driver of the manufacturing cost. We are also providing a full support, so scientific support to optimize your transfection process in the development phase, a regulatory support to get your drug approval, a supply chain to ensure the delivery date, and we are also providing GMP material, so with all the documentation. And we recently launched the regulatory support package, that is a documentation in which you will find all the information you have to know about our product. So it will also help you understanding how to build your IND and BLA.
So how can PILE also help you reduce the cost? There are several parts where PILE can help you do that. One part is certainly through our process optimization. So we provide our customers on-site support to better optimize and fine-tune their process and scale that up. We also have our accelerator process development services that can do monostep or end-to-end process development and or scale up for our customers. We also look at process intensification with innovative technologies and performance in order to decrease the cost. So our aim is really to help gene therapy drug manufacturers improve the cost per dose for better accessibility.
Right, so we would like to thank you for your attention, and we are able now to take any question you have. If we cannot answer during the live, feel free to send us an email. We will be happy to answer. I would like first to thank Emmanuel Camo to join me for this webinar, and I wish you a very good day.
Okay, great. So we do have a couple of audience questions for the speakers. So the first question is, do you recommend using a software to perform cost modeling?
Emmanuel, do you want to begin?
Sure. Emmanuel, you might be on mute still.
Sorry, yes. So you can certainly use a software, but you're not obliged to. I mean, you can easily also build model on Excel, and it can work provided you make the right assumptions and you have the right input data to put in. So certainly softwares can help in modeling these, let's say, facility data, but yeah, you can restrain yourself from doing that, and some people have built models that are published, like the work of Susie Farid from UCL, who have built a very extensive calculator, Excel-based.
To my experience, so for the Polyplus side, we made our own tool. And this is basically very good because it allows you to adapt the model to your need. So if you want to focus on a specific, for example, phase of the manufacturing process, so from our side, we focus on the upstream. We have the more detail for that, so we can adapt the level of details that we can have for this kind of thing. And it's also a very good exercise to learn more about the market. So to have a better understanding of the calculation, you understand the, let's say, the influence of each parameter that you modify. You control also the assumption. So as Emmanuel said, this is very important to build your own tool. But in the other hand, there is also some drawbacks. It's very time consuming. You need to update it I would say each year or each quarter, you have to have the resources to update it, of course, and it's also maybe a drawback as well. But the human errors in the calculation, it's always an issue as well. So with a software, I think it will delete this, let's say, drawbacks. So yeah, I would push for the handmade, homemade method because I like it, but that's my mind.
Okay. And when you show the cost of a plasmid, is it plasmids only or also reagents for transfection, other raw materials?
Yeah, so it was a plasmid only, basically, yeah. At least to my slide.
Yeah, and in my slide, it's complex, so it's transfection. Sorry, I think it's only pDNA as well in that model. So it's the pDNA cost.
Okay. And how sensitive is the cost of goods on the price of media and lysis components, if applicable? Yeah.
Emmanuel, do you want to take it?
So from what I've seen, the price of the media or the lysis components isn't the big part of the cake on the cost side. What is the biggest part is the pDNA and transfection reagent, let's say, complex. So, and this is why it's highly recommended to be optimized for an optimal cost. And also the depending of the model of production, it can be either people, so let's say labor, and then all the consumables that might be related to the process. But I don't see a big difference in using different medias on the overall cost, if that is what was the question.
Okay, so we do have a number of questions, and we are not going to be able to get to all of them today, but go ahead and continue to type in your questions. And also, just as a reminder, this webcast was recorded and it will be made available for replay, and you will get an email since you signed up for it. There's a number of questions about that. So the next question for our speakers is, so how should we approach cost modeling, and how do we validate our cost modeling model?
So I can start with that one. So I think it's the overall goal is not for everyone to start doing cost modeling, because it's a lot of time, of resource that you need to spend. So I think you can rely on your suppliers, such as us, to do it for you to some extent. And then, of course, but I mean, if the goal of a company is to build a special team and work on that, that's perfectly fine. But it requires a certain level of expertise and commitment to be able to really do it correctly. Related to, I forgot the second part of the question. So yes, how you validate? So to validate, I mean, I would suggest that once the model is built, you test it with a real process, and you see whether the outcome is makes sense. Like, for example, if you compare two different processes, whether the outcome is illogical compared to what would you expect.
Yeah, I support what you said. So the idea will be to be aligned on your purchaser, basically, and both for the upstream part and downstream part. So yeah, I would say it's important to have somebody who have the global knowledge about the pricing of the component that you purchase.
Okay, well, thank you, Emmanuel and Maxime, for joining us and presenting, and thanks to our audience. The recorded version of this webcast is available for replay in the multimedia section of our website. And as a member of our audience, you'll receive an email with a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye.
Bye.